Hi, I'm Nicholas Fernandez. I'm a computational scientist at the Human Immune Monitoring Center at the Icahn School of Medicine at Mount Sinai. And this tutorial is going to walk you through a short notebook visualizing the cancer cell line encyclopedia gene expression data using Clustergrammer2 on a Kaggle kernel. So this page is showing us a Kaggle kernel notebook where we are loading some cancer cell line encyclopedia data and we're going to visualize it. So in order to see the outputs, we're going to click the copy and edit button. So what this is going to do is begin executing the Kaggle kernel, which in this case is a Jupyter Notebook-like environment that is running Python and has many Python libraries pre-installed, and we've also pre-installed the Clustergrammer2 library. So this notebook is a short notebook where we've already preloaded the input data. You can see here the cancer cell line encyclopedia file here. So this is a gene expression data, and we're going to go ahead and set this one variable to true, the show widget variable, so that will allow the kernel to actually produce the widgets. And then we're going to go to run in the start top menu and run all. So while it's running, I'll explain what the notebook is doing. So the first thing we're doing is importing some libraries and just outputting what files we have in our input directory under Kaggle. So each of these cells can be customized, and users can change the commands to change the notebook. So we are importing Clustergrammer2, and it's giving us the version here. And then we're loading the data as a pandas data frame. So we can see the size of the data is 18,000 rows or genes and 1,037 columns or cell lines in this case. And we've pre-processed the CCLE data to add some categories of each cancer cell line. So the CCLE consists of 1,037 cancer cell lines, which are shown as columns here. And this is a bulk gene expression processing, and the genes are shown as rows. So although we have 18,000 genes, we're only going to show the top 1,000 variable genes across these 1,000 cell lines. So we're using Clustergrammer to visualize this large matrix, and you can actually see that we have a 1037 by 1000, so that gives us about a little over 1 million data points. And using Clustergrammer, we can zoom in and out of this very easily and see specific genes that are expressed in specific cell lines. And if we hover over the gene, it will give us information about the gene name and a refseq. And that information is coming from the Harmonizome web application from the Avimayan lab. So we've added some categories for the different cancer cell lines, and we can see the tissue here as the first category. So if you hover over a tissue, it'll show you the breakdown of where that tissue can be found. So we can see that the hematopoietic and lymphoid tissue cancer cell lines have very different gene expression than the other cell lines here. And we'll just move on to a different view where we're looking at exactly the same data, but we've now z-scored the genes across each cell line to show you how when you can transform your data and get a different view of your data. So this view highlights which genes are expressed relatively high or low in particular cell lines. So again, we see sort of the dominant clustering is this hematopoietic and lymphoid tissue, but other tissues also cluster in very distinct clusters. Like we can see a lot of breast cancer cell lines clustering over here. And Clustergrammer allows you to interactively explore your data in the sense that you can look at a cluster and see how that cluster is broken down into different cell types. So we can see this cluster is enriched for a particular tissue. We can order our cell lines, our cancer cell lines based on tissue and see particular patterns. We could change that ordering based on histology. We can also reorder based on parameters across the entire data set. So we can order our cell lines by sum to see which ones have higher and low expression, and then we can even pick a particular cell line and reorder all the genes based on that cell line, and we can see different patterns. So we have this sort of gradient of high to low expression across these different cell lines. And then we can, like we mentioned before, zoom in and see particular genes and just utilize this to perform exploratory data analysis of a large data set. And there's a lot more to explore, and we hope you enjoy the notebook.